Well, as I mentioned, I'm going to give you kind of a brief two-part presentation. So part of my current role as Executive Director of AOAC International, and this is a new program that I'm going to introduce a colleague who is leading this for us. And I think it's an important look, and it's just been a pleasure to be back at an ILSI meeting, which is something I've done very early in my career, and I was telling some folks last night, I think this is about the 30th year, so very excited to be back here. So just for those that may not be familiar with AOAC International, as I said before, we've been around since 1884. We were originally a part of the USDA. It was known as the Bureau of Chemistry way back. And when the Food Act produced what is now the FDA in the early 1900s, we were moved to the FDA, where we existed as an organization until the 1970s. And we were spun off as an independent nonprofit at that time and have operated that way since. When it was the thought, the motivation for that was that the science really needed to come not just from government, but from industry and academia, so that overall, collectively, we were relying on the best possible science to develop the standards and methods that the society really relies on for food safety and other things. So AOAC today, the acronym is the Association of Official Analytical Collaboration. And as we said, this work that we do has been relied upon by many individuals and regulatory bodies around the world to ensure sort of the fundamental parts of food safety, nutrition, and many other topics that have grown over time. So you see, we are a professional standards organization and scientific organization for analytical scientists and everything from chemistry, microbiology, genetics, lots of new roles have been created. So our vision, global alignment for trusted analytical solutions, kind of the ultimate, as Melanie alluded to, is to be getting a codex standard where harmonization to rule on trade issues and other things can be used. In our mission to advance food safety and product integrity through standards, validated test methods, and laboratory quality programs, and it's our compendium, the official methods of analysis, that has been around for over a centuries to develop all of these. So looking at the novel foods area, we have been staking this out as a while. There's certainly been questions that have come up, and my colleague, who I'll introduce here, and she's the lady in red, Kate Mastovska, is our Chief Science Officer at AOAC, and she's opened up a dialogue online just with a number of stakeholders that I think exceeded 100 over time, just asking about interest in this area overall. And just based on, this is not nationally representative or anything, but from the interest that we've gotten, the majority, 52%, are interested in really all the categories of cell-based, fermentation, insect, or plant-based. About 30% were interested in maybe some of the individual options, and just 18% were kind of interested in all the areas except for insects in that. So it gives you some sense that while we're kind of approaching this from a very broad aspect so far. So as Kate has organized our potential program in this area, she felt like the methods validated in novel food matrices needed to include routine analysis, reference analysis, authenticity analysis, or an advanced analysis area. So the first project that we're staking out, it's kind of a hard decision. Where do you start on something with a topic this big? Is on reference analysis, so the sum of amino acids for true protein determination. So the goal is to develop voluntary consensus standards for determination of total amino acids in selected food products and ingredients. And we have a development of what we call our standard method performance requirements, known as SMPRs, to serve as the basis for development of the AOAC official methods, which could be adopted by Codex Alimentarius. And we are putting together an advisory panel of supporters for this effort, and that panel would determine the matrices to be used in this initial work. So we certainly are looking for additional support. We would like to get this project kicked off this year. That is our goal, and we do have some commitments and been very happy to work with Stefan and ILSI, and I know we've included him in some of our initial programs with stakeholders. And I know he and Kate appeared recently together in a GIFSAN webinar on the same topic, so we look forward to continuing to work with ILSI on this. And so there you see Kate's email address along with mine, and there's also the QR code that you can go to get a little more information on this, because I've just given you the tip of the iceberg, and I hope that many of you will get a chance to talk with Kate on the breaks in the rest of this meeting. We'll be here until mid-afternoon tomorrow. All right, so now I'm going to switch gears real quick and pardon the deja vu for some of my friends who knew me with my IFIC hat over many years, but I found out that the IFIC colleagues were not able to attend this meeting, and I did get their permission to share some data that I had seen presented at the IFT meeting last summer in Chicago. So they've done a survey on innovations in alternative proteins, and this was a nationally represented survey of U.S. consumers that was done in May of 2023. It is weighted to the U.S. population with 95% confidence level, so certainly a very reliable survey of attitudes in the U.S. last year. So the highlights from this: more than half of Americans say they've tried an alternative protein at least once. General curiosity and a perception of healthfulness is the reason why they thought about trying it. And then in terms of typical consumption, about half of Americans consume animal meat daily, with taste, value, and general appearance being the top priorities when they purchase animal meat. And over 2 in 5 Americans would be interested in trying cell-cultured meat, while 51% say they would not be interested. Nearly 2 in 3 said that they would view meat products that are grown in labs using the cells of animals as very or somewhat safe if the USDA and the US FDA approve the safety of these products. And I think that makes the point that our keynote referred to earlier today, just how much value there is in that trust of the regulatory authorities. So, this type of result is really critical when we can align together and share, that it's not just one company's research or one university's research. You're talking about a great collaboration that can produce consumer confidence. And I spent a lot of time in my 23 years at IFIC dealing with that food biotechnology issue, and I can tell you there is a lot to be learned there, and the more we can get ahead of the issues, again, back to the keynote presentation, think about the possible ways that activists and others can use a topic like this to make it sound really unfriendly. So the more you can anticipate what those claims may be, the better you have a chance to answer with science that's accurate but also gives consumers confidence. Just to give you a sense of the definitions that IFIC used in testing these alternative proteins, you see they define cell-cultured meat and seafood is made through laboratory technology that allows for the production of animal meat and grown outside of the body animal cells from poultry or livestock. This development does not require an animal to be slaughtered in order for the product to be tested, to be created. Insect protein comes from specific insects that are grown and harvested for the purpose of human consumption. Mycoprotein, a protein source grown to have a meat-like texture from Fusarium venatum and a naturally occurring fungus that is edible. And then finally, precision fermentation protein is made with the use of microorganisms such as fungi or microalgae to make protein ingredients that are identical to animal proteins. So this is the last slide here. So what they found is the alternative proteins that garnered the greatest interest. Number one, cell-cultured meat at 42%, fermented protein at 37%, mycoprotein 36%, and insect protein at 29%. So again, based on reacting to those definitions that I just showed you. And I think what's quite interesting here is they've asked these questions back to 2021. So just 2 years earlier, the interest was only just about half that level. At 24% said that they'd be interested in cellular lab-grown protein, and only 27% said they were interested in fermented food products. So again, the word is getting out, and I think a lot more needs to be done, but I think this gives some really good food for thought as we go forward. So with that, happy to take a few questions, and all of the Daves are prepared to respond to those. So, thank you.
My question actually, since your first project is on amino acid analysis, and the first thought that came to me, will the methods distinguish racemic forms, and can you distinguish between the added and the natural, or are we going to be stuck with the same issue we have with sugars, where you can't distinguish an added from a natural in the label, and the FDA requires for the companies to be honest enough and disclose the added sugars on the label because the methodology doesn't exist. So is the form of amino acid going to be different, or the form of amino acid will be identical, that you're stuck with that same dilemma as sugar labeling?
Yeah, well, what I'm going to do is phone a friend on this. Kate had promised to answer any hard questions that came up on the data, but I think she can give some background, because we do think this project has benefits for traditional foods as well as alternative proteins, but I'll let Kate add to that.
So the starting point is to really have a good method for amino acids, because right now we don't have method for even general food analysis for amino acids. Your question is tricky, because like any added amino acids would be difficult to distinguish. So we would need to have very, I would say, potentially advanced methodology, for example, using on like isotopic ratio, if you're using, for example, not natural sources to synthesize, amino acids, right? So that would be like a very advanced and quite expensive testing. So it might be possible in the future, but right now our focus is really even on the basic stuff, like having actually reliable method for amino acid testing, so we can actually start comparing different products when it comes to the profiles of amino acids, and also have a reference method for example, like a fast methods like Kildare or DIMA, where we're using the nitrogen to protein conversion factors, which right now are not developed for novel foods. I'm not talking about the cultivated meat, I'm talking about like insect-based or some, let's say, plant-based proteins, which might be unique and may not have these factors yet. So we basically need a reference method now. So I would say we are pretty rudimental, at this point, what we actually need.
Hi, thank you. I'm from ILSI Taiwan, and I'm also associated with AOAC Taiwan. Okay, my question is about the food allergen. Okay, when the alternative protein come from the fungal, microalgae, insect, and probably a lot of people never touch those proteins at their whole life, then it becomes alternative protein source, and how we can evaluate, or any technology we can detect in possible allergen inside that product. Thank you.
Maybe I can jump in with that a bit. It's very difficult to predict if a novel protein is going to become an allergen, and people don't really know the reasoning behind it. But across the biological world, there's a lot of protein similarities. So that the way that it's normally done is there's a rolling window of 88 amino acids, and if you have a significant amount of a homology between your new protein and something that's an existing allergen, it will be flagged as an allergen. And it doesn't necessarily mean that every person who is allergic to that protein would be also allergic to the novel protein in question, but it's a possibility. So then you can flag that with labeling and things like that, and people can know to avoid it or undergo an oral antigen challenge or something like that. But that's really the best way that we have to do it now. People are working on new techniques to determine whether a novel protein will cross-react with existing allergic IgE, for instance. But we're not quite there yet.
I have another one over there. Yes.
Hello, I am H.N. Misra from India. So I have been listening this very carefully, and my one concern is, or rather I would like to be educated about the commercial aspects of the technology, commercial production. Is there any data available or some study has been done somewhere that for large scale manufacturing of this live grown meat, what will be the infrastructural facilities required? For example, suppose we want to grow 1,000 pounds of this meat. So how much time it will take? Or what is the minimum, that is the cost required for producing this product that is a economically viable production technology? Can you throw some light on this? Thank you.
Yeah, no, I mean, as far as there being a commercial data, I mean, there just isn't. Right now, I mean, Upside, for example, they're currently just starting. They broke ground on their commercial facility in Illinois, I don't know, a few months ago, and I mean, that's, you know, they're still, again, building that. And as I mentioned before, I mean, as far as the economic aspect, I mean, yeah, it'll be at a high price point more than likely, but the goal is to make it as comparable or even cheaper than your conventional products. So, I mean, that eventually is the goal. But directly to your question, is there data right now? No.
The success of the technology, that is very important. That is how to take it to the market.
Oh. Yeah, Dave.
Yeah, I could just add a little, because we had a whole symposium last week on this. I think the general feeling for the group that was there, academics, industry, government, investors, companies, everyone was there talking about this. And everyone agreed generally that there's, you know, the infrastructure doesn't exist. And that's a lot of cost. And companies, startup companies, can't afford to build these plants. So it's going to take government contributions, whatever you want to call it, or significant government inputs, whether it's in India, whether it's the US or elsewhere, to build the plants and build a facility so the companies and others can use them, start to produce at the scale. So I think a lot of it depends on when those investments happen. And I think the technology, the science, the kind of regulatory safety, that's all coming along, but at some point you're going to need a real bolus of support to build, you know, the hard, you know, the foundations, the production factories and so on. That's not going to happen fast unless there's an influx of funding. That's the opinion I would give. Okay.
So one over here.
Yeah. Have you got any perspective on like halal or kosher certification on these products?
Ooh, no, not. Maybe one of the regs can.
Only speaking from what I've read in the news, but one of the leading countries in production of cellular agriculture is Israel. And I had read that one of the chief rabbis of the country was expressing interest in finding a way to declare the products made there kosher. I don't know much about the process that goes into that, but I know that because Israel is definitely a world leader in the field, and so it's of great interest to them, I believe. Yep.
Yeah, and I believe they just got their beef product approved a few days ago. So yeah, certainly.